Hello, my name is Joshua Mgavi. I welcome you to Cell Respiration. How do cells obtain energy for their functioning? Cells obtain energy for their metabolic reactions from breaking of organic molecules with high energetic content. This energy is almost stored as ATP molecules. The process of obtaining energy in order to produce ATP molecules is named cellular respiration. What is the compound that is phosphorylated for ATP formation? What is the resulting compound when ATP liberates energy? ATP, adenosine triphosphate, is formed after binding of one phosphate, phosphorylation, to one ADP, adenosine diphosphate molecule. This is a process that stores energy into the produced ATP molecule. When ATP gives energy to cellular metabolism, it loses one of its phosphate and ADP reappears. ADP can also lose more phosphates and generates adenosine monophosphate or even non-phosphorylated adenosine. Adenosine production from ATP is a solution used in tissues that need urgent oxygen supply. For example, the heart during myocardial infarction, or what we call heart attack. Since adenosine has a local vasodilator effect, thus providing faster vasodilation than other physiological methods. What are the types of cell respiration? There are two types of cell respiration: aerobic cell respiration, a reaction with particular molecule of oxygen; anaerobic cell respiration without participation of molecular oxygen, but with other inorganic molecules as oxidants. There are several varieties of anaerobic cell respiration. The main one is fermentation. Under which conditions do aerobic cells carry out fermentation? Some cells that usually obtain energy from aerobic cellular respiration can carry out fermentation when oxygen is not available. They are bacteria and fungi that, under the absence of oxygen, use their anaerobic metabolic capability for energetic supply. Muscle cells carry out fermentation too when oxygen is scarce. What is the difference between aerobic and anaerobic beings? Aerobic organisms are those whose cells do not survive without oxygen, since they depend on aerobic cell respiration to obtain energy for ATP production. Anaerobic organisms are those ones that live or can live under oxygen-lacking environments. What is the difference between facultative anaerobic being and obligate anaerobic beings? Facultative anaerobic beings like fungi, Saccharomyces cerevisiae, brewing yeast can survive under oxygen-poor environment, carrying out fermentation. However, when oxygen is available, these beings can carry out aerobic respiration. Obligate anaerobic beings are those that cannot survive when oxygen is present. Some fungi, some bacteria, like the agent botulism, Clostridium botulinum, and the agents of tetanus, Clostridium tetani, and some protozoans are examples of obligate anaerobes. What are the two types of fermentation? What are their chemical equations? The two main types of fermentation are alcoholic fermentation and lactic fermentation. Alcoholic fermentation, pyloric acid, an intermediate molecule, is converted to ethanol with libation of carbon dioxide. The alcoholic fermentation equation is as follows. Glucose, there will be phosphorylation of 2 molecules of ADP with inorganic phosphate to give us ethanol plus carbon dioxide and 2 ATP molecules. Lactic fermentation, the pyloric acid is transformed into lactic acid. There is no production of carbon dioxide. Lactic acid fermentation equation is glucose, 1 molecule, 2 ADP molecules with inorganic phosphate to give us lactic acid and 2 molecules of ATP. In general, What are the reagents and products of fermentation? In fermentation, glucose, the sugar, is degraded into pyruvic acid. Each glucose molecule forms 2 pyruvic acid molecules. In this process, 2 molecules of ATP are produced. According to the type of fermentation, pyruvic acid can produce ethanol and carbon dioxide, that is in alcoholic fermentation, or lactic acid in lactic fermentation. There are other varieties of fermentation in which pyruvic acid can generate acetic acid, acetic fermentation, propionic acid, isopropanol, and alcohol too, and many others. The type of fermentation depends on the species of the involved organism. Why in cake and bread manufacture are alcoholic fermenting organisms used, not lactic fermenting organisms? Fermentation has a function of making the cake and bread grow. This is accomplished by libation of carbon dioxide in alcoholic fermentation. As the gas passes through the dough, it makes it to grow. In a lactic fermentation, there is no libation of carbon dioxide, and the desired result would not be obtained. To what substance is the acidic flavor of fermented milk due? Some bacteria ferment milk lactose by lactic fermentation, producing lactic acid. This product is responsible for the acidic flavor of yogurt, curd, and milk. How can the knowledge about fermentation explain the origin of muscle cramps, pain after intense physical exertion? A typical fermentation process due to oxygen scarcity happens in the muscle tissue. Under intense use of the muscle, demand too much energy, ATP, and consume much more oxygen to produce energy. High consumption leads to oxygen scarcity, and muscle cells begin to make lactic fermentation, trying to satisfy their energetic needs. In this situation, muscle pain, cramps, and fatigue are due to lactic acid released by fermentation. How many ATP molecules are produced from each glucose molecule used in fermentation? How many ATP molecules are produced for each glucose molecule used in aerobic respiration? In fermentation, from one glucose molecule, 2 ATP molecules are produced. In aerobic respiration, much more productive process. From one glucose molecule, 36 ATP molecules are made. Which cell organelle that is specialized in aerobic respiration? The cell organelle that is specialized in aerobic respiration are the mitochondria. Of which main compounds is the mitochondrion structure made? Mitochondria are organelles delimited by 2 lipid membranes. The inner membrane invaginates to the interior of the organelle, forming cristae, and delimiting the internal space known as mitochondrial matrix. What are the 3 phases into which the cell respiration is divided? The 3 phases of aerobic cell respiration are glycolysis, Krebs cycle, and respiratory chain, also known as electron transport chain. What is glycolysis? What are the products of this process? Glycolysis, the first stage of aerobic cell respiration, is a process in which glucose is degraded or broken to form 2 pyruvic acid molecules, along with the formation of 2 ATP and 2 NADH. Glycolysis is a complex reaction implying the formation of several intermediate molecules until pyruvic acid molecules are made. Although 2 ATP molecules are consumed in the reaction, there is also production of 4 molecules of ATP. Thus, a positive balance of 2 ATP molecules is obtained. Two NADH molecules are also produced. In glycolysis, the 6-carbon structure of glucose is broken to 2 organic chains of 3 carbons. Each are made. These chains give birth to 2 pyruvic acid molecules. Does glycolysis occur within the mitochondria? Glycolysis happens in the cytosol, not within the mitochondria. Pyruvic acid molecules later they will enter the mitochondria to participate in the next phase of aerobic cell respiration. How many ATP molecules are made after glycolysis? Glycolysis is a process similar to glucose degradation in fermentation. It produces a final balance, 2 molecules of ATP for each broken glucose. What is NAD? What is the role of the NAD molecule in glycolysis? NAD, nicotinamide adenine dinucleotide, is a hydrogen acceptor necessary as reductant to receive hydrogen in the same reaction, in which it is reduced and converted into NADH2. During glycolysis, 2 NAD molecules retrieve hydrogens liberated after the intermediate reaction, thus forming NADH2. What happens during aerobic respiration to pyruvic acid molecule made by glycolysis? What is the sequence of reactions that then follow? Pyruvic acid molecule made in the cytosol by glycolysis will enter into the mitochondria. Within the mitochondria, each pyruvic acid molecule is converted into 1 molecule of acetyl-CoA, enzyme A, with the liberation of 1 carbon dioxide. The Krebs cycle, also known as citric cycle, the second stage of aerobic respiration, then begins. What is the official name of pyruvic acid? Pyruvic acid is 2-oxopropanoic acid. It is thus a molecule made up of 3 linear bound carbons with 1 extremity forming an organic acid functional group, and the middle carbon binding to an oxygen atom by a double bond. Why can it be said that each glucose molecule runs the Krebs cycle twice? Each glucose molecule cycles the Krebs cycle twice because after glycolysis, each used glucose has generated 2 pyruvic acid molecules, and each pyruvic acid is converted in a 1-to-1 proportion of acetyl-CoA. Each acetyl-CoA then cycles the Krebs cycle once. Why is the Krebs cycle also called the final common pathway? of the degradation of organic compounds. The Krebs cycle is called the final common pathway of degradation of organic compounds because it is also possible to generate acetyl-CoA from the degradation of lipids and proteins. Since acetyl-CoA is a substrate that triggers the Krebs cycle, this process is called the final common pathway for being activated by other organic molecules, those are lipids and proteins, and not only by glucose. Organisms use energetic reserve of fat and proteins to cycle the Krebs cycle when undergoing malnutrition or when there is no glucose available for the cell. What are the final energetic products of each round of the Krebs cycle? Where is the most part of the utile energy at the end of the Krebs cycle found? After each round of the Krebs cycle, 2 carbon dioxide molecules, 8 protons, those are hydrogen ions, captured by NAD and FAD, hydrogen acceptor, and 1 ATP molecule are produced. During the Krebs cycle, acetyl-CoA is degraded. At the end of utile energy is incorporated into hydrogen transported by FADH2 and NADH2 molecules. How many carbon dioxide molecules are liberated at each cycle of the Krebs cycle? For a single glucose, how many carbon dioxide molecules were already liberated by aerobic respiration at that point? Each round of Krebs cycle liberates 2 carbon dioxide molecules. At the end of the cycle, all carbon atoms from the original glucose molecule degraded in glycolysis are already liberated, incorporated into carbon dioxide molecules. That occurs because for each glucose, 2 pyruvic acid molecules were made by glycolysis. Each of these 2 pyruvic acid then is converted into acetyl-CoA with liberation of 1 carbon dioxide molecule, 2 in total. Since each of the 2 produced acetyl-CoA cycle, the Krebs cycle once from the initial glucose 2 rounds of the Krebs cycles is generated, and so 4 other carbon dioxide molecules are made. All the 6 carbons of the glucose molecule are then incorporated into 6 carbon dioxide molecules, 2 made during acetyl-CoA formation and 4 during the 2 cycle of the Krebs cycle. Where in mitochondria does the process called respiratory chain occur? Which are the products of the Krebs cycle used in that final phase of aerobic respiration? Respiratory chain, or the electron transport chain, is performed by protein systems located in the inner membrane of the mitochondria. Energized electron of hydrogen atoms transported by NADH2 and FADH2 are the products of the preceding phases used in the respiratory chain. What are cytochromes? Cytochromes are proteins of the internal mitochondrial membrane that are specialized in electron transfer and participate in the respiratory chain. Energized electron liberated by hydrogen donors, NADH2 and FADH2, reconverted into NAD and FAD, pass through a sequence of cytochromes, losing energy in each passage. The energy is then used in the synthesis of ATP. How is the respiratory chain? How in the respiratory chain do electrons from FADH2 and NADH2 passing through the cytochromes liberate energy for the ATP synthesis? What is this ATP synthesis called? FADH2 and NADH2 oxidate into FAD and NAD and liberate hydrogen ions and highly energized electrons in the beginning of the respiratory chain. The energy lost by electrons that pass through the cytochromes is used to pump protons, that is hydrogen ions, out of the inner mitochondrial membrane to the region between the inner and the outer membrane of the mitochondria. Hydrogen concentration gradient between the inner and the outer space, delimited by the inner membrane, forces protons, hydrogen ions, to return to the mitochondrial matrix, the region inside the inner membrane. However, that return is only possible if hydrogen ions pass through an enzymatic complex called ATP synthetase embedded in the inner membrane. In that passage, ATP synthetase phosphorylates ADP and then ATP molecules are produced. Hydrogen liberated in the mitochondrion then combines with the oxygen to form water. As the reaction that depends on oxygen, this type of ATP synthesis is called oxidative phosphorylation. Until the Krebs cycle, aerobic respiration can be described without mentioning oxygen, the chemical element after which reaction gets its name. Where in the process does the chemical element take part? What is its importance? Oxygen enters aerobic respiration in its final phase, the respiratory chain. It is fundamental importance because it is responsible for the maintenance of the hydrogen concentration gradient between the space separated by the inner mitochondrial membrane. This gradient promotes the functioning of ATP synthetase and thus phosphorylation of ADP to form ATP. In space inside the inner membrane, oxygen binds to free hydrogens to form water. And this hydrogen consumption keeps the hydrogen gradient and proton traffic through ATP synthetase. The entire aerobic respiration process has the intent to make ATP synthetase work. Aerobic beings, for example, we humans, need to breathe oxygen to maintain the hydrogen concentration gradient and keep ATP synthetase working. How does poison cyanide act upon aerobic respiration? Cyanide is a poison that inhibits the last cytochrome of the respiratory chain, interrupting the ATP formation and thus leading to the cell to death. What is anoxia? Anoxia, this is a situation in which there is no available oxygen in the cell. Without oxygen, the respiratory chain stops. There is no ATP production. The cell does not obtain energy and dies. Anoxia can be caused, for example, by pulmonary insufficiency, for example, drowning, extensive pulmonary injuries, and many others, by obstruction, halts, and deficiency in tissue circulation, atherosclerosis of the coronary arteries that irrigate the myocardium, tunicates, heart arrest, by hemolysis, that is lysis of the red blood cells, or hemoglobin disease, anemia, fetal erythroblastosis, and many others. How many ATP molecules are made after aerobic respiration? And what is the net energetic gain of the process? After aerobic respiration, 38 ATP molecules are made with the consumption of one glucose molecule. But 2 of these ATP are consumed by glycolysis. So the net gain of the process is then 36 ATP molecules per glucose molecule. What is the general equation of the aerobic respiration, also representing ADP and phosphate? The general equation of aerobic respiration is 1 molecule of glucose plus 6 molecules of oxygen, which I'll get, plus 36 ADP and 36 inorganic phosphates. We get 6 molecules of carbon dioxide, 6 molecules of water, and 30 molecules of ATP. Why can the consumption of molecular oxygen indicate the metabolic rate of aerobic organism? Molecular oxygen consumption has direct relation to cell metabolic rate in aerobic cells. So to the metabolic rate of the organism, cells having higher metabolic activities demand more energy. Such energy comes from ATP molecules. As there is need for ATP production, the intensity of aerobic cell respiration is also higher, and then more oxygen is consumed. Thank you very much for participating in cell respiration. Please do like, share, and subscribe.